Hello everyone, welcome to Organic Chemistry 1 Lab Experiment 5. Today we will learn how to perform TLC analysis. The first step is to prepare 5 samples. We have 4 standards here and 1 unknown. The unknown code is A. We will dissolve a small amount of sample with ethanol in a small test tube. I have 5 spatulas for 5 different samples because we don't want any contamination. No need to measure your sample. You just need to estimate the amount with your eyes. We only want a very small amount, so this is enough. Can you see the white sample on the bottom of the test tube? Now it's time to add ethanol. The liquid in this beaker is absolute ethanol. We want to add 1 ml of absolute ethanol. However, no need to measure the volume with a granted cylinder, because it doesn't have to be exactly 1 ml. We will transfer ethanol with a dropper, and we can see the 1 ml mark on the dropper, so we can estimate the amount with this dropper. And if you have any sample attached on the inner wall of your test tube, try to rinse it off. Make sure all the solid is fully dissolved. Okay, please take a look at it. Now it's a clear solution. And then make sure you label it. There are 5 different samples. However, after dissolution, all the samples are clear solutions. If you don't label your test tubes, there's no way to distinguish your samples. The first sample I made is salicylic acid, so I will label it with SA. Before you use a plate, please check it under the UV light to see if there is any contamination. If the plate is already contaminated, don't use it. Draw a straight line with a ruler. Again, please make sure you do it with the ruler. This is the starting line of our samples, and it must be higher than the solvent surface in your TLC chamber. Draw some dots on this straight line. The number of dots depends on how many samples there are. We have 5 samples, that's why I drew 5 dots on this line. Those 5 dots must be evenly distributed on this line. I labeled those dots with 1 or 2 letters, and we will spot our samples exactly on those dots. S stands for salicylic acid. AC stands for acetaminophen. C stands for caffeine. AS stands for aspirin. And U stands for unknown. Now let's see how to spot your sample onto your plate. Dip your capillary tube into your sample. Now can you see the liquid? This is your sample solution. We call this one fill. First, finish one fill on your plate. Then check it under the UV light to see how it looks like. If it's not enough, we can spot more. Your sample looks like a circle on your plate. You want to try to minimize the size of the circle. The liquid in this container is absolute ethanol. To clean your capillary tube, you want to dip it in ethanol, take it out, and dry it with a piece of paper towel. You want to repeat this process at least 4 times. This is how the plate looks like under the UV light prior to the development in a TLC chamber. Please pay attention to the colors of our samples. These 4 samples are purple, while salicylic acid is kind of pink. Can you tell the difference between those 2 colors? This is our TLC chamber. I made it with a beaker and a watch glass. The purpose of this filter paper inside is to accelerate the formation of the equilibrium. You only need a small amount of solvent. As I mentioned earlier, the surface of your solvent must be lower than our starting line. Otherwise, your samples will be dissolved in the solvent. Now let's place the plate into our chamber. Try to keep it vertical. Can you see the solvent going up on the plate? Now it's time to take it out. To save your time, you don't have to wait until the solvent has reached the top edge of your plate. Once about 85% of your plate is covered by the solvent, you can take it out. You want to mark the solvent front immediately because the solvent disappears very fast after you have taken the plate out of the chamber. Now let's examine our plate under the UV light. Don't worry about the color here around our solvent front. This is actually caused by the solvent. Can you see a spot here? It's kind of purple and kind of pink. This one is salicylic acid. The next one here is acetaminophen. The third one is caffeine. The fourth one is here. This is aspirin. The last one is our unknown. According to this TLC plate, are you able to figure out the identity of our unknown